Okay, okay. Thank you, Suchapa, for the kind introduction, and thanks to the organizers for giving me this opportunity to talk about the development of PDB-Dev, a prototype system for archiving integrative structures. I thank the speakers who, JP and Dina, who kind of gave an introduction to what is integrative structure determination. But just to continue in that direction, what is integrative structure determination? Integrative structure determination is an innovative approach to modeling the structures of biological systems based on data produced by multiple experimental and theoretical methods. It is typically used for modeling the structures of complex macromolecular assemblies that are not amenable to traditional methods of structure determination, such as X-ray crystallography, NMR spectroscopy, and electron microscopy. Integrative modeling involves combining data from multiple experimental methods such as X-ray, NMR, EM, small angle scattering, cross-linking, mass spectrometry, Forster resonance energy transfer, electron paramagnetic resonance spectroscopy, etc. The experimental data are gathered and converted to spatial restraints and combined with known starting structures of molecular components using computational modeling algorithms to build the structures of complex macromolecular assemblies. Before I go into the details of archiving integrative structures, I'd like to give some background information regarding the Protein Data Bank and how the project of archiving integrative structures began. PDB is the single global repository for experimentally determined structures of biological macromolecules and their complexes. It was established in 1971 with 7 structures. It is the first open access digital resource in biological sciences and currently contains over 170,000 structures of macromolecules. The PDB is managed by the Worldwide Protein Data Bank organization that consists of the RCSB PDB in the US, Protein Data Bank Japan, PDB Protein Data Bank in Europe, and the Biomagris Bank. The PDB archives structures of macromolecules determined using X-ray, NMR, and 3D EM methods. The structures archived in PDB have atomic coordinates and metadata. PDB also archives certain structures that are determined using multiple methods. These methods usually use one of the traditional methods such as X-ray, NMR, or 3D EM in combination with other methods such as neutron diffraction or small angle scattering. And these multi-method modeling result in structures in atomic resolution. Here's a plot of multi-method structures archived in the PDB over the years. And you can see that multi-method structures have been increasing in the past decade. So the next question is, can the PDB archive integrative structures? There are some technical challenges to archiving integrative structures in the PDB at present. These challenges arise due to the following factors. Integrative modeling involves restraints from multiple experimental methods such as chemical cross-linking, small angle scattering, FRET, EPR, etc. Integrative models can be multi-scale, which means that they're not necessarily atomic and can consist of both atomic and coarse-grained representations. Integrative models can also be multi-state ensembles comprising of constitutionally and conformationally diverse assemblies, and they can use starting structural models that come from experimental or computational methods. Due to these reasons, the current PDB infrastructure is unable to handle integrative structures. So in order to address the challenges involved in archiving integrative structures, the Worldwide Protein Data Bank set up a task force consisting of researchers from different fields contributing to integrative structural biology. The first meeting of the task force took place in October 2014 at EBI in Cambridge. Here's the summary of recommendations from the task force that was published in 2015. The task force recommended that structural models, data, metadata, and workflows need to be archived. A flexible model representation needs to be adopted. Methods need to be developed for assessing model uncertainty. A federated network of archives need to be created for structural models and experimental data. And publication standards need to be established for integrative modeling studies. In this talk, I will focus on the implementation of the first two recommendations, which is archiving integrative structural models, data, metadata, and workflows, and adopting a flexible model representation. With this background information, we can state our objectives as follows. Our goals are to build a system for archiving integrative structures based on the recommendations of the WWPDB Integrative Hybrid Methods Task Force, and to develop the tools required for archiving integrative structures. And while building the archiving system, we will adhere to the FAIR guiding principles of scientific data management, where integrative structures will be findable, accessible, interoperable with other data resources, and reusable for future applications. So what are the requirements for archiving integrative structures? The first requirement is the creation of data standards. Data standards provide the foundation for archiving and enable automated data processing, as well as dissemination of data in a standard form for downstream applications. Data standards primarily encompass definitions for data that need to be archived, such as the chemistry of macromolecules and small molecules, and the definitions for multi-scale model coordinates, along with some additional metadata information about the data that has to be archived. The second requirement is the archiving system. We need to build the infrastructure required for data collection, processing, archiving, and data distribution. Data collection involves collecting the model coordinates, the associated spatial restraints, the starting models used, as well as modeling protocols. Data processing involves any added curation that may be carried out, model validation, as well as visualization of integrative structures. Data distribution involves creating mechanisms for web services, data download, search and report tools, etc. Together, these constitute the data pipeline for the archiving system. Let's first look at the first requirement, which is the creation of data standards. We have created the IHM Data Dictionary, which is an extension of the PDBX MMSIF Data Dictionary. PDBX MMSIF is used by the PDB and provides the data standards for archiving atomic structures of macromolecules determined using X-ray, NMR, and 3D EM. In the IHM Extension Dictionary, several standard definitions from PDBX MMSIF are used. These include definitions for the chemistry of macromolecules and small molecules, atomic model coordinates, and supporting metadata, such as information regarding source organisms, samples, authors, citations, etc. And the new definitions that are required specifically for integrative modeling are created in the IHM Extension Dictionary. The data standards defined in the IHM dictionary can be outlined as follows. The details of molecular components in the structure, such as proteins, nucleic acids, and small molecules, and the corresponding references to sequence and chemical information databases. These are taken from the PDBX MMSIF dictionary. The definitions for starting models used, along with references to models that may be available from the PDB Model Archive or PDB-Dev. These are provided in the IHM Extension Dictionary because these are specific for integrative modeling. Similarly, the input spatial restraints derived from experiments such as cross-linking, SAS, FRET, etc., along with references to data residing in other experimental data repositories such as PDB, BMRB, EMDB, SASDB. All these are also captured in the IHM dictionary as these are specific for integrative modeling. Together, these are applied to build an integrative model, which can be any combination of multi-scale, multi-state, ordered ensembles. Multi-scale models correspond to models that have both atomic and coarse-grained representations. Multi-state models correspond to models in multiple conformational or constitutional states. Ordered models correspond to models that belong to a time-ordered path or models that belong to another ordered path, such as a biochemical pathway. And ensembles are collections of models that represent the solution set. So the IHM Dictionary provides a flexible model representation that can handle multi-scale, multi-state ordered ensembles. The IHM Dictionary is open source and is available from GitHub. Next, we move on to the second requirement, which is the creation of an archiving system. We've developed PDB-Dev, which is a prototype system for archiving integrative structures. The data standards for PDB-Dev are provided by the IHM Dictionary. PDB-Dev data collection and current procedures. We've developed a simple web interface for depositing structures to PDB-Dev. Structures can be submitted at this link, which is also available from the PDB-Dev website. An MMSIF file compliant with the data standards in the IHM Dictionary is required for data submission. The current processing procedures include ensuring that the submitted data are complete. During the curation process, we add reference sequence information from, say, UniProt and GenBank. We check for compliance with the IHM Dictionary. We visualize the structures and then issue accession codes. We also allow for structures to be kept on hold before publication. Visualization of integrative structures. There are two applications that currently support visualization of integrative structures, the Chimera X desktop application and the Molstar web application. Both these applications support the visualization of multi-scale structures with atomic and coarse-grained representation. And Chimera X also supports visualization of multi-state structures and also some of the spatial restraints that are used in the modeling. This image corresponds to the integrative structure of the 552 subunit nuclear pore complex from budding yeast archived in PDB-Dev and visualized using Chimera X. This model was obtained using spatial restraints derived from 3D EM maps, chemical cross-linking data, and small angle scattering profiles. PDB-Dev data distribution, we have a newly redesigned dynamic website. This is the link. The website provides functionality to search and retrieve archived structures in PDB-Dev using keywords. There are also individual entry pages for each entry that provide additional information regarding the structure itself, the author's citation software, as well as input data used in the modeling, along with links to data in other repositories. Model coordinates can be downloaded from the website, and models can be visualized on the website using Molstar. This slide shows a snapshot of some of the structures archived in PDB-Dev. The structures vary from small and medium-sized complexes such as the BB-Zome and the mitochondrial iron-sulfur complex to very large complexes such as the nuclear pore complex, the BAF complex, and the RNF ring domain and nucleosome complex. This slide shows the current status of PDB-Dev as of December 2020. There are 55 structures that have been released and 10 additional structures that have been processed and are kept on hold for publication. The plot on the left here shows the statistics for the software used to obtain PDB-Dev structures. Several structures have been obtained using the integrative modeling platform software, Rosetta and Haddock, and there are fewer structures from other modeling applications such as FBS, PatchDoc, BioEN, etc. The plot on the right shows the statistics for the types of experimental data used for modeling structures archived in PDB-Dev. Chemical cross-linking is predominantly used, followed by electron microscopy, NMR, SAS, FRET EPR, etc. With regard to software tools, we've developed the Python IHM software library to support the IHM dictionary. This library has been developed by Benjamin Webb in Andre Charlie's group at UCSF, and it provides a mechanism to programmatically generate MMSIF files compliant with the IHM dictionary. It can be used as a standalone program or as part of modeling applications such as IMP or Haddock. The Chimera X software also uses the Python IHM library. The software is also open source and available on GitHub.
30 more seconds, Brinda.
Sure, thank you. So what are we currently working on? We're working on developing methods for model validation as recommended by the WWPDB-IHM task force. We are also working on building the infrastructure for automated data harvesting. We need automated tools for data harvesting because users have model and restraint data in different formats that do not allow any data standards or that do not follow any data standards or data specifications. So we are trying to create an automated system for harvesting diverse and heterogeneous data. Future perspectives, our goal is to build all the tools required for archiving integrative structures. Once the mechanisms for collecting, processing, validating, and archiving integrative structures are fully established through PDB-Dev, these structures will be moved into the PDB archive, and the tools will be integrated with the tools in the PDB. Another long-term goal is the creation of a federated network of interoperating resources that contribute to integrative structural biology. This goal poses additional challenges such as development of common data standards and tools for federated data exchange. We conducted a workshop in 2019 to address these additional challenges for creating this federated network. If you're interested in learning more, the outcomes have been published and you can look at the paper. I'd like to thank my mentors at Rutgers, Helen Berman, Stephen Burley, and John Westbrook, who have provided invaluable guidance and support throughout the project, and my colleagues Kathy Lawson and Mariam Fayazi, who helped with the PDB-Dev website. Our collaborators at UCSF and USC, Andre Charlie's group is working on developing methods for model validation, and Carl Kesselman's team is helping us with the development of the data harvesting system. I'd like to thank the developers of Chimera X and Molstar for developing the visualization tools, the WWPDB directors for their support, my colleagues at RCSB PDB, and also all the PDB-Dev depositors for providing us with examples and working with us patiently while we build the system. I thank all the members of the WWPDB task force for their continued guidance and support. And finally, I'm grateful to the United States National Science Foundation for providing funding for this project. Thank you all for your kind attention. Please visit the PDB-Dev website for downloading or depositing structures. I'm happy to address any questions.